This is a video about our Comsci project. This is Shanuga, and that's me, Thalerni. And we decided that our group name would be undecided because we took forever to actually decide a name. And we decided that our idea of a project was going to be basically on biology, and we called this Project Biology 101. So initially we would have a DNA sequence, which is then transcribed into mRNA and then translated into proteins of many functions. DNA is typically made out of nucleotides, which are represented by A, T, C, and Gs. mRNAs are also made out of nucleotides, but these are T, U, C, G. Meanwhile, proteins are made of amino acids. In any given cell, when we have DNA sequence with nucleotides, the A nucleotide gets converted into mRNA as U, while the T nucleotide becomes an A, the C nucleotide becomes a G, and the G nucleotide becomes a C. A codon is defined as a sequence with 3 nucleotides. So any mRNA sequence is split into 3 nucleotides each consisting of a codon. And these codons specify for a specific amino acid. So there are altogether 21 different amino acids, while there are 64 codons that encode for them. For any given functional protein, the mRNA sequence must start with methionine, which is AUG, while in the DNA sequence it would be written as TAC, and it must also end with one of the 3 stop codons, which are UAG, UAA, or UGA, and in DNA sequence that would be ATC, ATT, or ACT. Without the start codon, you won't be able to process the mRNA sequence, and without the stop, you won't be able to get a finished protein product. So this is Mr. Hocus Pocus, and he introduces you to his class of biology, where he's going to teach you how to convert any DNA sequence into proteins. And you're given instructions where you have to create a sequence with A, C, G's, T's, and it has to be within 6 to 50 characters, or you could say bye to leave the classroom. So for their first sequence, we give a TAC, AGA, AAA, CAG, and ACT. So the TAC is the start codon, the ACT, which is at the end, is the stop codon, and there are 3 other codons, the AGA, which goes for serine, AAA, which for phenylalanine, and CAG for valine. So our program runs the sequence, and they say hocus pocus. He uses magic to show methionine, serine, phenylalanine, valine, and stop, which was as expected. So for the next sequence, we will use a sequence without, so the same sequence that we use, but without the start codon. This would be AGA, AAA, CAG, and the stop ACT. And this would be run to show the same protein as before, because in our program we have an automatic adding of the methionine. For the next sequence, we decided to put just the start protein and a codon and then an incomplete codon, and this would produce a non-functional protein, which would be only methionine and serine. The next sequence we tried was with numbers and non-nucleotide letters, and this tells you to follow the instructions. And then if you enter BYE, the professor basically leaves the program and you exit the program. So the whole point of this function is that you set the error to false, and we use this idea from term test 2 question 2. So if set error is false, found error to false, you would run through the whole sequence until you find an error or until you have gone through all the characters in the sequence. If there is a character in the sequence that is not supposed to be there, that would change the found error to true. And once found error becomes greater than 1, so it's true, or there are multiple errors, then you have another function which can decide between two things. If found error is greater than 1, that would mean that there's an issue with the sequence, so you would read it. If it says bye, then initially what they want to do is that they want to exit the program, and so we have the effect of Hocus Pocus leaving the classroom. Whereas if it has letters and numbers, then you tell them to follow instructions and ask them for another sequence. If there are no errors found, then you could run the sequence properly. The other two functions included effects, which brought upon Hocus Pocus, and then the wand, which was able to rotate.
The process of converting a DNA sequence to a protein is divided into 4 functions. The first function converts the DNA sequence to an mRNA sequence. Using an index, the function will go through each letter of the sequence entered by the user and will add the corresponding complementary nucleotide to the list mRNA sequence. For example, if letter index of DNA is A, then the letter U will be added to the list of mRNA sequence. The index will then change by 1, and the same process is repeated for the next letter until the index is greater than the length of the DNA sequence entered. The list mRNA sequence is then going to be used in the next function. In this function, we've also included an if statement that will incorporate the nucleotides for the start codon in the event that the user doesn't include it themselves when entering a sequence. If the first 3 letters of the DNA sequence entered aren't the nucleotides needed for the start codon, the function will add the complementary nucleotides to the list mRNA sequence. Since our code aims to mimic the actual process of protein synthesis, which can't occur without the start codon, we felt it was necessary to include this. Moving on to the second function. The second function groups the mRNA sequence into codons. A codon is a group of 3 nucleotides, so this function essentially groups the mRNA sequence into groups of 3. Similar to the first function, the function uses an index and goes through the list of mRNA sequence. Letters index, index plus 1, and index plus 2 of the mRNA sequence will be added to the list codons, then the index will change by 3. This process is repeated until the index is greater than the length of the list of mRNA sequence. This function also includes the block mutations, which looks at mutations in the sequence entered. The mutation function looks at 3 possible errors in the sequence entered. 1. The length of the mRNA sequence is not divisible by 3. 2. The sequence has more than one stop codon. And 3. The last item of the list of codons is not a stop codon. If any one of these conditions is met, the function will say that a non-functional protein will be produced. To see if the number of stop codons is greater than 1, we use an index and set the item index of the list of stop codons to equate to each of the 3 stop codons. If the item is one of those stop codons, then the counter variable number of stop codons will increase by 1. This process is repeated until the index is greater than the length of the list of codons. The next if statement looks at if any one of the 3 mutation conditions, which have been incorporated in the form of booleans, has been met, and will say if a non-functional protein is produced or not. The last function converts the list of codons into a list of amino acids. Like the first 2 functions, we use an index, and using if statements, see if the item index of the list of codons is equal to a specific codon. If it does equate to a specific codon, then the corresponding amino acid is added to a list of amino acids. In this function, there are 20 if statements to account for the 20 amino acids that are possible from the codons. Once the corresponding amino acid has been added to the list, the index changes by 1. The function will continue this until the index is greater than the length of the list of codons. At the very end, the function will say the list of amino acids as the protein sequence.